My name is Nathan Cady. I'm an assistant professor here at the College of Nanoscale Science and Engineering, and my specialty is nanobioscience. The instrument is a very interesting instrument. It's similar to atomic force microscope. So what it uses is very small cantilevers, kind of like a fountain pen, to be able to print very small spots of fluids on surfaces. Now you might ask, why would we be interested in printing small drops of fluid on surfaces? Well, for a lot of biological problems, especially developing things like biosensors, we're interested in spotting proteins, DNA, and other biological molecules at specific locations on some of the devices that we produce. What we're really talking about is the interface between biology and either a detection device or a device that might be helpful for medical applications. So you might imagine things like implantable devices where you might need to interface with cells. This could happen with neuron research, where you want neurons to grow in specific locations, or interface with electrodes. This type of work can help us pattern chemicals or molecules that might attract those cells to specific locations on a chip that we might implant. In addition, we may actually work with developing sensors where we want to detect a lot of different targets at one time. This tool allows us to spot specific capture molecules that can bind to either cells that we're interested in, viruses, or even some of the DNA and proteins that are associated with those pathogenic organisms, in order to identify them and find out if they're in a sample or maybe even in a person.